Okay, thank you folks for joining us for our final honors program colloquium for the fall 2022 semester here at A&M Texarkana. My name is Craig Nakashian, and I'm the director of the Academic Honors Program, and I am very, very excited to welcome my once and former colleague, I suppose, and friend, Ben Neuman, who is giving his annual virus lecture for us here at A&M. Dr. Neuman is a professor of biology and chief virologist of the Global Health Research Complex at Texas A&M University down in College Station. And this year's annual holiday virus lecture is entitled Fun with SARS-CoV-2 and Spider-SARS. So you're going to tell us all about that. We should have some time for questions afterwards. Otherwise, if you could keep yourselves muted, then please keep yourselves muted until we are done. All right, thank you. Dr. Neuman, take it away.
Okay, will do. There we go. All right, hide the little pictures over here, and we're good. All right, yeah. Hello. So I wanted to start out with a little, this is what's going on in the world right now thing, because I figure that's probably your most immediate interest, and then I can show you some cool stuff at the end. So here we go. All right. So there's the big present. Yeah. Were you good this year? I hope you were. Anyway, so what did we get? Well, we got an early Christmas this year in that flu arrived early. So the little lines are cumulative flu cases in, different flu seasons. So you see sometimes it's high, sometimes it's low, but usually they all start coming up right around week 50, 51 of the year, which is right at the end of the year. This year are the little red dots. So you can see we're not particularly big. We're only kind of medium-sized or small in terms of flu, but look where it's happening. It's starting about 10 weeks early, maybe 12 weeks earlier than it ought to be starting. And that is what the problem is. So flu, it's a big problem. People are talking about it. The people who are worried about it are saying, well, compare it to last year or any other year, and it's this big and they're all that big. But I would say it's probably just a normal sized peak that's coming early. What else is under the tree, though? Let's see. Oh, my gosh. Look at that. Yeah. RSV. So this is the one that babies are getting. Older people can get it as well. It's like a cold that'll just sit there. It can linger for a week. It can linger for a month. Normal time of year is like January, would be the peak of RSV, but look at that RSV peak. So it seems to be going up and maybe coming down, which is good. But here again, it is way too early. Yeah, a couple months earlier than it ought to be. And the third thing, I'll dispense with the stupid presents, is COVID season. And COVID season is really just getting started. So over the last 2 weeks, cases in Texas doubled compared to the week before Thanksgiving. So right on schedule. Yeah, we did this, pat ourselves on the back. It's coming. Interestingly, everybody's talking about flu, but if you look, there are consistently somewhere between 8 and 12 COVID deaths per flu death in the US. And there have been, recently. And this is peak flu. So peak flu is 1/10 as severe as the lowest COVID that people have seen in a long time. That is generally taken as a good sign. Other neat thing, yeah. If you know 12 people who died last year, one of them died with COVID. If you know 15 people, one of them died solely of COVID. Yeah, 1 out of 15 literal deaths in 2022, which was considered not the bad year. This is after we'd, stopped it and COVID was over. Yeah. But still, yeah, it's about 8.5% of all deaths in the US. Yeah, which is really saying something. I think, though, that the season thing, like this idea that we can always prepare for the next COVID wave, probably doesn't really hold much water. If you look at us, and then I put the UK data down there, they've had 4 distinct, very high COVID peaks this year, one right after another, about as soon as you could possibly imagine. And a lot of their peaks come right where our trough was. They have most of the same traditions. They're even starting to get into that old Black Friday stuff. They're really just missing Thanksgiving, but I don't think we can pin it on the weather. I don't know that we can pin it on the time of year. COVID has basically thrown everything out of whack so that there isn't really a season for most of these viruses that used to have a season in the past. And so, yeah, the bad thing is this is all influenza-like illnesses. So your COVID, RSV, flu, colds, whatever. And as you can see, we're peaking really early, but we're also peaking really high. So this is everything all together, and we're running with these three sort of outbreaks at once, and that's why they're saying, oh my gosh, everybody wear a mask for Christmas, which I don't know that anyone will, but that is probably the only thing that kind of blunts this very sharp upward trajectory at the moment. I guess RSV is coming down, but RSV is a tiny, tiny fraction compared to COVID cases and flu cases right now. So there we go. All right. So you also probably want to know, all right, well, which one's going to get me? Yeah. And so these are the ones that are going around right now. It's the same sort of offenders. If you're in Africa, it's probably going to be BA4, which is weird because that one has gone extinct everywhere else. Everywhere else, it's going to be one of these guys, and they have fancy new names, but they're all just Omicron, really. And what I mean by that is here, look. So this is a list of shared mutations. It's actually a full list of mutations. And everything at the top is an Omicron. And so when they have something in the same column, that means it's the same mutation across what we consider multiple strains of the virus. Basically, all the Omicrons are the same. They're all one thing. Down here, the bottom half, these are all the old variants. Then you've got your alpha, your beta, gamma, delta, kappa, and they're all really the same thing. And the giant panda over there is waving goodbye because all the ones at the bottom are extinct now. We have driven these things over the edge of the cliff. Yeah. And they're gone. I think we can probably do it. There's only been one major shift in COVID. So all these other little tiny variants kind of don't matter because if you can vaccinate against one of these things in this group, you probably hit everything else in that group. So the problem is, we've got 300 and I forget how many, like 330 million people in the US, and we've only distributed the new updated booster to 39 million. So we're about 12% vaccinated against Omicron, which means we're about 88% not vaccinated against Omicron, which means the holiday season is a great big open door decorated with tinsel and lights and all that stuff, welcoming COVID in, basically. Yeah. And if you wanted to see, well, how we doing here in Texas, you pick your part of Texas, but it's one of the browner states, brown being bad and green being good in this map that I think is friendly for colorblind people. But how do we know this works? Well, we got 70% of the US vaccinated with at least one dose against the original 2019 version, and 2019 version is gone. Now, add that on to the roughly 20 or 30% of people that definitely had the early version of COVID, and everybody's got some immunity to that. It's just that there are so many changes in Omicron that basically it's like starting over. And even a lot of the people who got sick last January were still catching Delta, the last end of the Delta. So something like half of the people that think they're immune to Omicron have zero immunity to Omicron. And anyway, if it was a year ago, you probably need a booster anyway. They don't seem to last much longer than 6 months. So that's where we are. Yeah, that's why everything's bad. Okay, so I don't know. What are we doing? What's anybody doing? So I want to show you what I'm doing, and let's translate it into real people terms. So, all right, so we got these little chemicals in the corner, protease inhibitors. If you know, like Paxlovid, the Pfizer drug, that's a protease inhibitor. We've also got this other class of drugs. They're called PROTACs. They're pretty neat. They basically distract the virus and then call for help. It's like a sort of a two-pronged attack. And then we've got ritonavir, which basically stops our things from getting degraded. So if we were translating those into other terms, we'd say, all right, the protease inhibitors are kind of like a monkey wrench. We're throwing it at the virus, but they're also designed so that they will actually bind forever. Yeah, so that's why the ring is there. They stick to one molecule from the virus and they kill it forever. It can never let go, which is really cool. Paxlovid doesn't do that. This new kind that we're working on do, and they seem to work pretty well. Low side effects, really nice, everything so far. So yeah, it's like a terrible marriage in that they're bonded forever and nothing works after that point. The middle one is, I was thinking it's like a kick me sign, but it's more like a panic button because it's distracting the virus over here. One end of it looks exactly like the protease inhibitor. The other end binds to the cell's protein destruction system. It's called the ubiquitin system. And they basically tag these proteins. And after that, the proteins get destroyed. It's really cool. The third one that makes our drugs last longer is similar to, I don't even know what. Yeah. But yeah, this is what I imagine it's like. And it does. It really prolongs the effect for more pleasurable, virus inhibition. So why are we using these protease inhibitors? What's a protease? Like, why would that even work? Those are good questions. So here is an explanation. So this is the machine at the core of the coronavirus. It just looks like a bunch of squiggles because that's how proteins look. But each of these colored bits is doing a separate job and they're interacting with each other. You got to imagine them moving around as they're working. The pink thing coming out of the center is a new copy of the virus. So this is the virus copying machine, the thing that makes all that COVID stuff possible. All right. So these come as one big long protein. And the first thing the virus does is it cuts the protein into little parts so that they can go and make different parts of the machine. And so, for example, the two green circles here actually were attached to the green circle over here. This is, the thing that the protease cuts apart. And once they're apart, then they can go and form, the correct version of this. And so yellow, there's the start, there's the end. Orange, that one's pretty close at least. Here's the red, here's the purple. So basically, yeah, look at that one. If these two aren't cut, this end is attached to that end, and that is not going to make a machine that looks right at all. It's going to be cramped and awful, and it's probably not going to work. So when we take out the protease, we're taking out the part of the virus that cuts at every single one of these spots. And so it is a way that you can actually stop literally every other part of the virus from doing anything, which is what I think makes it so fun. Yeah. Oh yeah, 1415. All right. So basically what we're doing is bullying. We are here to bully the virus. We are here to beat it down, curb stomp it, etc. That doesn't look right. That's better. Yeah. Viruses, not people. That's the way you do it. Yeah. I went online. I found a stock photo of bullying. I thought this was worth sharing because look at that. It's called Kick of the Bully. Oh my gosh. Yeah. From iStock. Look, the bullies are smiling. They're nicely dressed. And it looks like she's prodding a dead body with her toe. I just think, well, if that's what bullying is, I don't know. I need to turn it up a little bit. Here are some of the drugs. So this is what we're going to be looking for. So the lines here, when we're looking at like the red line, let's focus on that one. What we want is more bang for our buck. I mean, isn't that what anybody wants? So you can only kill the virus till it's dead. I mean, that doesn't even need to be said. But the question is, how much of your drug does it take to kill the virus? And does the virus get back up off the floor after you knock it down? Those are the things we worry about. So the little red one, let's say, is one of our drugs with the solid triangles. And so that's fine. That's pretty good. The blue one we would say is a better drug because it takes less of the drug, so less over here, to get you to the same point. So our floor is about there versus about there. And so basically you want to look for curves that are shifted to this side, and then those are going to be the good ones. The purple drug over here is another one we're testing out. It's the one that works by pushing the panic button. It is not great at its job, but it has some interesting properties. So I don't know, we'll still go. And I don't know if you did much bullying back in your high school days or elementary school days, but the key to good bullying is keeping useful statistics because you need metrics. You need to know, whether you're connecting, whether you're doing the right thing. So in our virus bullying, our chief metrics are called the EC50 and EC90. And that's the amount that it takes to knock out 50% of the virus versus the amount it takes to knock out 90% of the virus. Yeah. Now, this plus RIT over here, that is our little purple pill, basically. And so what you see is without RIT is the white square. With RIT, look at that, it gets more effective. Actually, this little purple pill makes everything more effective, shifts all the curves. So it's pretty useful. And this is one of the things that they put in. So the blue one is the Pfizer pill, that's Paxlovid. And they actually mix it with the ritonavir, the purple pill. And so the solid blue squares are the best you can get from the pharmacy at the moment. So now, with that in mind, if that is the best we can get from the pharmacy, the blue line, this is the new drug that we've got. And it's way better. Yeah, it's like, I don't know, 20 or 30 times more bang for buck. And the really cool thing is that we can mix it with a number of these not so effective drugs, the kind of wimpy also-rans. And this is it with nothing added. So this blue line over here. And as we add each of these drugs, even ones that do not work well at all, they gang up on our poor little virus and just absolutely maul it. It's really wonderful to see. The three most interesting drugs that we threw at it are over here. They work in different ways. It's like having bullies, punch from different sides. You don't want to be doubling up on the same spot. Your drugs will get all tangled up. So it's important to diversify. And yeah, the 3-drug cocktail is a heck of a lot better, something like 10,000-fold better than the best drug that is out there on the market right now. So yeah, that could be good. Yeah. We have to go into some animal and people trials, but those are coming. We got some funding for it. So yeah, let's see. All right, next thing, fun with lasers. So we've got some of these proteins from the virus. We want to see how they work. The way all these proteins work is they stick together. And the thing is, you can't see a protein. You can't see either of the proteins. And so it's really hard to tell if they're stuck together or not stuck together. But that's how your drug binds to the target. That's how antibodies bind to the virus. It's how basically everything that we want to happen happens. So we came up with this cool way with Raman microscopy that actually works. So what you do is, step one, you shoot the virus with a laser. And I know we talked about that last year, but the same laser, it's a good thing. Shoot the virus with a laser. And then one of the things that happens is that it will set the virus to vibrating, or rather it will set all the molecules in some parts of it to vibrating, just like this. And these are what they call distinct vibrational modes. Which one is your favorite? I feel like that's just like a morning stretch. That's okay. The scissoring looks like it's asking for help. Wagging is just a little too in your face with the two coming towards you at the same time. I'm a fan of rocking. I think that's just sort of a nice, happy looking thing. And twisting, it's kind of high energy, but right time, right place. I could totally see that being a thing. Anyway, each of these vibrational modes can happen to some extent. Each one eats up a different amount of energy. And so you shoot in light of a particular energy / color, and you'll get light back out of a different energy / color. And the difference between those two, they can actually, pin down and tell you exactly what happened. So the way we use this is there. We can take all the components that make up a virus protein or make up any protein. So here are the individual amino acids. We can shoot them each individually with the laser and see, okay, if we have this amino acid, we're going to have a lot of peak over here, not so much over here. We can take all these little curves. We can feed them to an AI because, my brain can't handle this stuff. And we can say, okay, we're looking at a protein that should be about this big. This should be its spectrum, and we can get actually a pretty good match. We can even take an unknown protein, zap it with a laser, and say, wow, there's probably 5% tyrosine, 2% tryptophan, et cetera, down there. And we're still training the AI, but it seems to be working well. But the other thing that happens is when two molecules stick together, they basically stifle the party. So all those molecules that were wagging and just going nuts are now stuck because they have another molecule next to them. And so generally, you're going to knock down some of these peaks when you have a molecule that is stuck to it. And so if you know what either of the molecules looks like, you can say, oh, wow, when I put these together, can we see some sort of change? And the answer is, yeah, you can see a real nice change. So here are just one way of looking at the changes. These are SARS antibodies. And so when we put in antibodies that don't bind at all to our virus, they end up right over here. So control IgG, those are goat antibodies that have never seen SARS in their life. So they're absolutely useless. When we put in antibodies that don't bind, they end up actually right on top of those. It's the darker dots. They are mixed in with the gray. There is no difference there. We put in an antibody that binds, but real weakly. It's sort of barely hanging on by a toenail. That's our green pile up here. And when we put in antibodies that actually bind usefully, cover up the spike, and would prevent infection, we get a giant shift all the way over to here. And so you can put in the little target from the virus, put in your antibody, and you can tell whether or not this thing is actually going to work. Would work against any strain, work with any antibody. And it's kind of neat. It's a way you can see something that you can't see with your eyes, but can be absolutely certain of it. And so we can do this with antibodies. We've done this with molecules binding to proteins. We've done it with proteins binding to proteins and changing shape. It's, it's going to be very interesting. I have a feeling this is going to be big, but I don't know how big yet. We'll see. Yeah. Okay, last bit, little Spider-SARS bit. So like, where does all this mayhem that we're dealing with right now, SARS-CoV-2, fit in? Well, so I think I talked at some point in the past about some of these relatives of coronaviruses that we found. We've finally got a good handle on where these things sit. And so we've got group names for them. We got the Archaea viralis, because archaea sounds a bit like arachnid, and every one of those is in a spider so far, or some kind of arachnid. Those are the first branch. So this is where we connect to other viruses, and the first branch of the tree, boom, arachnid viruses. Second branch, we're calling the Protonata Viruses. These are all in insects, except one is in a mite, which is a kind of arachnid, and that one is the first branch in this little part. So we've actually got these Protonata viruses branching from basically within Archaea virus territory, which is cool. And what you see is that all these little branches are of similar length, similar degree of evolution, because all these viruses have a problem. If you look at them and line them up, not one of these can really change. They all have all the same parts, and all they can do is drift. So it's basically like being in a canoe without a paddle on, I don't know, the Red River when the water's low and nothing's flowing anywhere. You just drift. And that is not a good way for a virus to evolve. So we started out with these things that were very bad at evolution. And then we make the jump and look at the branch lengths go. So longer branches mean more changes that the virus was able to pack in. They all start at the same point in time, but these guys win the race. So these are the nidoviruses. These are the ones that include SARS. And I marked up where some of these things are coming from. So they're roughly lined up with that tip. Early branches, all in arthropods, mostly in the water, but yeah, crabs and shrimps and things like that. Next branches, mostly arthropods, but a couple other weird things you find floating in the water and little flatworms. Then you get to the cluster that includes SARS-CoV-2, and we are sandwiched between a virus of an axolotl, giant Mexican salamander, and a virus from a fish. And the one that's upstream of us is a virus of a frog, and the one that's downstream of us is a virus of a different ocean fish. Yeah. We are a weird little coincidence, just this strange virus from a whole great big spectrum of viruses that happens to have really taken hold and, yeah, honestly, really done a number on the world. And so you can see a couple of times the virus group seems to have been able to merge over into mammals. And so those are the little pink boxes that we have here. The one we really don't want to get is this box at the bottom. So that's this one that's got the monkey on there. That's basically coronavirus Ebola. It causes massive hemorrhagic fever in the monkeys. It is highly lethal. Yeah, really bad stuff. So far, it doesn't infect humans, but it's close. It's infecting lots of different primates and doing terrible things to them. So that's one that I worry about personally. I think it's almost cracked the code, and I hope it never finishes. Yeah. So here's a look at some of their genomes, and I color-coded all the same parts in the same color. And these are the Protonata viruses. And honestly, the most different thing about them is that they have different names. Everything else about them pretty much stacks right on top of everything else. They have all the same parts in all the same places. Yeah. Because they're basically stuck. All they can do is drift. And so what do I mean by parts? Well, here, yeah. So these are proteins again. This is just what proteins look like. You don't have to worry about them. That's just who they are. Each of the different colored bits is going to do a different part of the job. This is basically that same complex from before, but we're just looking at a kind of slimmed down version of it. It doesn't have all the parts in there. The important thing here, though, is what do they do? Like, what's their role? And so here's their role. Like, imagine we're making a wedding dress, a beautiful wedding dress. Yeah, this is a wedding-themed talk, or relationship-themed, I guess. Yeah. So we've got one protein that kind of serves as the mannequin. This is the thing on which we are going to build the rest of the virus. And there's a special part, and all it does is add the very first, basically like the very first stitch. And so boom, now we've got one little bit. From that point, there's a big part that will take over and it'll make all the rest of it. Just copy, copy, copy. Then, as you do, we have a separate part, and it's the same part that was working up here. It's going to take our completed virus off the scaffold, and then we can reuse the scaffold again, just like a mannequin. It's kind of neat. Now, there are other virus parts, and they're going to do the other job. So we've got one that holds everything in place, like little pins, one that kind of works like scissors. It cuts away bad nucleotides, things that you don't need. One that will help that one by unzipping. So if you make a mistake, or if the virus makes a mistake in copying, it'll pull it back. And this is the little zipper that separates the good copy from the trash copy. And it feeds the trash copy to the pair of scissors, basically. It's like throwing a food scrap to the dog under the table. Yeah. We got one that flattens out the RNA coming in, and we got others that add the little bells and buttons and bows that just make everything work. But yeah, so if that's what a coronavirus has, which parts do we find on these other viruses? Because these are the earliest branches, and a virus will always change anything that it can change. And so if we can find anything that stays the same, one, we may be able to understand it, and two, if we can stop the old version, we may be able to stop the new version. At least this is the theory. So here's what we've got in the new viruses. Very little and a lot of questions. We've got the big part that's going to complete the job. We have none of the pieces that ought to be starting the job. That's very worrying. These viruses are littler, and so maybe they're just more tolerant of mistakes, but they do not have a mistake-correcting pair of scissors in there. And they also don't have the little unzipper that goes with it, but they do have the thing to flatten out the genome before it gets copied. So I don't know. And then also, even though this has to work in the same way, like a normal wedding dress, we are missing every single one of the components that would be turning it into something you can actually wear, that basically turn the virus into something that actually works. So yeah, very strange. We do have the little pins, though, the bit that holds basically the iron part onto the sort of main working part. And so it looks like coronaviruses start out with just this very small ensemble. And then, like any good factory, as they evolve, they pick up new pieces, and they don't put them in the middle of the assembly line. The middle stays right there. They add them onto the front end, and they add them onto the back end, and they add them on around the assembly line. Because we're just changing the finish, we're changing, little tweaks and things, but the main process is still the same. So if you want to see the result of this, at the top, we've got our viruses. These are the archinidos and the protonidos, and they're very similar. This is just one region of the genome. Compare that to the absolutely mind-bending diversity that we get in the rest of the coronaviruses. The only thing that they all have in common in this region is the blue bit, which is the main protease. Yeah. And that's the thing that we've been making protease inhibitors against. So what we're going to try is we've got several of these ancient proteases, and we've got them cloned up, and we're going to sort of boot them up. Just a little part of the virus. We're not creating new viruses from the past or anything crazy like that. Yeah. But the idea is that we've got now a panel of about 100 fairly good drugs that seem to work against SARS-CoV-2, like the COVID protease. And so the idea is, if we can find something that hits the ancestors, that hits the old ones, that'll probably be hitting some part of the protease that is the virus is unable to change. And where the virus can change and try and squirm out of one of these, if we can go in and hit it with with one that controls like the core version of it, and hit it with another one that really suppresses it, then potentially we can bully more effectively. The protein from SARS is shown down at the bottom, not rotating, because my student is good at that and I am not. But you can see they've got the same thing. They have these two little twisty barrels and then some helices out the side. Yeah, so they're all basically the same thing. So I'm kind of excited to see what we find from that. All right, and so to end, here's a little Christmas recipe. Apparently this came from some goofball at Ohio State. You just take a styrofoam ball or one of those little flower foam things and spray paint it gray, spray paint some cloves red, stick them in pointy end first, and you got yourself a little COVID Christmas ornament. You can pass them out to all the people that you don't like and don't ever want to come back to your house. It's just a nice way of saying, stay away. Yeah, COVID's here. I think that's enough from me. Thanks very much, for this.
Okay. Thank you, Ben. I appreciate it. Yeah. You can put up your little applause if you want, or not. I don't know. Register your disapproval of the sad and annoying message from Dr. Neuman.
Don't encourage me. Thank you. But yeah.
I love the ornament though. That's fantastic. And it's cloves, so it's Christmassy. But of course, yeah, my cats are growling at each other because that's what cats are supposed to do here in this most ridiculous of timelines we're living in. So, all right, I'll open it up to questions. I have a couple, but I will defer, not very long, until to anyone else. So if you have a question, you're welcome to unmute and ask it, or if you want to drop it in the chat, I can dramatically read it. It's up to you. Well, now I have a cat that's coughing. All right, well, I'll ask a question then whilst we wait. Unless you were going to, Corinne. Were you ready?
I'm thinking about it, but I do have one. I just
Okay,
have to figure, I just need to articulate it correctly.
You think on that. Now I need to remember mine, unfortunately. Yeah, my first one was in some of those drug cocktail things you were talking about, and some of the work you're doing. I know that this is not binding or anything, but for the populace, what's the time to market on something like that usually? You are muted though, Neuman. Again, we've only been doing Zoom for 2 years.
Do you remember when people used to Zoom bomb because they had to be part of the Zoom? Yeah, Zoom was the place to be, and you just had to force your way. Yeah, a simpler time. So repeat that question. It went completely out of my mind.
What's usually the time to market on something like that?
Oh yeah,
we've been pushing as fast as we can, but
with a couple $billion dollars behind you, you can get through all of this stuff in about 3 months and have it ready to be in human clinical trials. And then it's something like a year to get through those trials, but a little faster if you're Pfizer, because they are good at paperwork. Yeah. Okay. Putting together bits and pieces and passing around my little cup. It's a good question how long it'll take, but they seem promising anyway.
Okay, well, that's good. A bit of good news in the otherwise bleak holiday COVID season.
I think it depends how good an industrial partner we're able to get. We've got one right now that's not really doing much with the patent. And if we get somebody that's got a little oomph behind him, then I think, maybe it'll go faster. Yeah.
Okay. Did you have your question ready, Dr. Hinton, or should I ask my second one?
No, I'm typing out in the chat so that I have it in front of me. Okay, so given that flu arrived, 10 weeks or so earlier than normal, and RSV has already peaked, if I look at the sort of other layers of how RSV is active, should we then expect them correspondingly to wrap up a bit earlier this year, or are we just looking at a longer, more sustained season of giving from both of those?
What a nice way to put that.
Thanks.
question.
I was just taking your presence metaphor and running with it.
Yeah. RSV normally just does one peak and then it's gone. So that I'm at least hopeful that we're looking at the back of that one. Flu in a normal year comes up and then it sits at the top. It just sort of zigzags for about 3 months. And so if it's doing that, it's going to go over the entirety of holiday-induced COVID season. And that is going to be real mean and real bad. I'm hoping it doesn't. Last year it didn't. It was just one little peak and then it came straight back down. But I don't know. I don't even know anymore. Yeah. So I am not hopeful. Yeah.
Excuse me. Do we have
I
any?
want to say thank you, but I don't feel like I should say thank you for
None
that.
of us
That's
want
not the right response. Yeah.
Grim Reaper of joy every time
That's
we
right.
have these
Yeah,
talks.
sure. A middle finger is fine, honestly.
No.
You remember when we started doing these in like 2017, and it was just a bit of fun around the holiday to talk about viruses, but they never
sure.
affected anything.
People like viruses. Kids are attracted to them. Yeah, I understand. Yeah.
What are the again, other questions? By all means, drop them in the chat or unmute or put your hand up or whatever. But in terms of, I mean, obviously there's a fair amount of gloom because that's the world we live in. But there was some very hopeful stuff that you talked about in terms of some of these treatments and the lasers and such like. So I guess the question I would have, I want to be a little more positive, is what would what do you see as the potential implications beyond COVID for some of these treatments? And, can we hope to maybe have a little optimism that they'll be effective against other perennial nuisances?
Right.
Feel free to say no. No, they won't.
No, I think yes. Yeah, I'm glass half full, maybe more. Yeah, probably all the way full. Yeah. No, so we're kind of at the dawn of what I would call, so the Dutch concept was total football, like where every player on the soccer team can play every position and you just do maximum soccer and somehow that makes you win. Unclear on all the other details of it, but World Cup I think is going on. I don't even know.
The Dutch did just beat up on us, Ben. Thanks.
They did. Sorry, too soon, maybe. Yeah. But we can basically start to go total football on coronaviruses. And that's not something we could do before because the existing inhibitors hit different parts of the virus. And we're trying to come up with a couple more of these complementary inhibitors. Basically, there's no virus that can be held down and effectively bullied by 1 or 2 compounds alone. It looks like you need a minimum of 3 pushing it in different directions. That's what has really been a game changer for HIV. And so I think we're at the dawn of that. Now, with HIV, what you see is that you have cocktails, but each company does their own one with their own 3 drugs. So it took a lot longer for each company to build up to the magic number of 3, at which point they could make the cocktail. So we're still kind of there, and we're trying to dump a whole great sort of brain load of these things onto the world at the same time. So hopefully they can be picked up. And we're also looking at these as enhancers for some of the other drugs that are out there. Because, yeah, it's good to go into bullying with a friend. Yeah, basically.
Okay. So, yeah, I guess it just goes without saying that hopefully the drugs that treat COVID have also been improving alongside the vaccines and such, which raises a whole another weird social science mentality of people who choose one or the other, but I'll take them all. Any other questions that we've got? I see Sydney wanted to have a question to hear me dramatically read it, but I'm sorry, Sydney, you need to have a question for Dr. Nakashian to read. And I will, I'm stalling. I will put, that's not what I wanted.
I'll unmute. I got one.
Thank you, sir.
For what's the greatest danger for folks who are doing everything right? They're masked up in public places. They're vaxxed to the max. They're wearing their mask in crowded places. What's the greatest danger for that crowd?
JeanPaul Sartre, COVID is other people. Yeah. Yeah, basically. Yeah, it's the people that you let over and let in, unfortunately. Nobody can be a chain by themselves, and there are some pretty weak links out there. And yeah, that's what you got to watch for. And I don't even know that you can watch for it because they're just all around us, all over the place.
Yeah, but if you're taking
yeah.
precautions in public crowd in the, crowded places, elevators and all that, then
This is good, but
Yeah.
there's probably somebody in your life that you don't take precautions with. And so that's the way the virus gets in most likely. It's the unguarded moment, or it's the other person, or the other people that come in. And there's not a great way to knock out all the risk, unfortunately. Yeah.
I'm hearing an analogy with the safe sex education.
Yeah,
You're
why
inhaling
not?
every colleague your partner ever inhaled.
That's right. You're breathing every virus. Yeah, your guests have ever inhaled, kind of in the last couple of weeks anyway. Yeah. Yeah, unfortunately. But I like that people are
That's all
still
right,
trying.
Right.
So
And
Ben,
the-
I have a question. Maybe it's anecdotal, and you can tell me whether or not this is true. In the last few folks that I have known that have had COVID, including myself back in August, I noticed that this time around, having been a second timer, this time around, it took more days and more tests and being a more persistent self-tester to test positive. And I've had that same experience accounted for, and a couple of other folks were like, well, I thought maybe it was COVID, but I tested negative, so that's not it. But then they tested again 2 days later and they're like, oh, well, there it is, right? Are the current variants or strains taking a bit longer to increase that viral load to where the sort of self-tests are popping positive? Or is it that we're just more aware and hypersensitive to any sniffle or sneeze or scratchy throat might be COVID, so we're testing sooner, but then I think in some ways alleviating ourselves and saying, oh, well, it's negative, so it's not COVID and I can walk around the world. And it's like, well, it is, you just don't know it yet.
Yeah, you're probably right. So I can answer your anecdote with an anecdote. Why not? When our family caught COVID over the summer, everybody got it. And it was probably my fault. Yeah, which is terrible. Yeah. We were testing and we were sequencing and I filled up all these little jars full of human saliva, banked for later because it's got the good stuff in there. Yeah. And what we found out was that at least in the 4 cases in our family, we got very little in the nose. The nose swabs would come up negative most of the time. The one swab that would reliably come up positive was a saliva swab. And so we would end up just sort of licking the end of the detector and then shoving it up our nose, because otherwise, I guess that's more gross if you do it in the opposite order. But that was highly successful. We basically figured out we would have missed about 90% of the days we were positive if we had just used those rapid home tests and only used them with the nose sample. But if you use a saliva sample and you're doing the PCR test, it's like 10,000 times more effective at picking it up. So yeah, partly test, partly site. And it did seem as if this last version of the virus didn't grow as well in that little part of the nose, which is the thing that we're sampling. Those up the nose things, that's an old flu test. And it only works to the extent that COVID grows there, which that's not really its home. So it was kind of a lucky coincidence at first, but now, yeah, we're missing all kind of stuff. So better to
should
put
we be
mouth.
sticking the self-test swabs in our mouths and doing them with saliva rather than up our noses?
In your mouth or up your butt. Yeah,
Oh,
those would
okay,
both
well,
actually
I'm
be.
gonna
I
go
was
with the
just saying, yeah.
former and not the
Saliva is
latter.
fine. Yes.
I also
yeah,
think my 8-year-old would appreciate the former and not the latter.
Likewise, I would plan on doing the former. Yeah. Myself.
Make sure
Just
you get
giving
the
people
swabs.
options. Yeah.
That's good. Very helpful for my humanities students where we just watch some bureaucracy
Yeah.
when they do the hospital testing. Any other questions? And yeah, there's the soundbite for CNN. Thanks, Ben. Stick it up your butt, you're fine. Excellent. This is nothing but the finest. Any other questions for Dr. Neuman while we have him? I did put the link to the honors program YouTube channel over there. I guess they changed the little URL. It actually has our name in it now, which is fascinating. So very exciting. You can also find his earlier his earlier videos from April of 2020, I think December of 2020, December of 2021, and December of 2022. And I might go back and watch them all in sequence to see the evolution of our understanding of COVID. Although I think your April 2020 video probably still holds up. I mean, I think it's, we had a pretty good handle on it all the way back then. And all right, any other questions? I'm gonna call that no. All right, in that case, thank you again, Dr. Neuman. As always, I would say it's a joy to talk to you, but it is a joy to talk to you, even if the subject matter is a bit bleaker. If you ever want to talk about like mushrooms or fossils, let me know. That's a little cheerier. I was gonna say, who am I asking? We'll do it in the next hour. Yeah. Let's just keep going. Otherwise, have a good semester, everybody, and you too, Ben. Appreciate it. And yeah, thank you. Have fun.